Role of abscisic acid in plants. A major function of abscisic acid in plants is to cause stomata to close during water stress or when elevated carbon dioxide levels occur in guard cells in the evening when photosynthesis stops but respiration continues. 2. Waring and his colleagues, who led to the discovery of dormant abscisic acid, showed that levels of this compound increased considerably in leaves and buds when bud dormancy occurred in the short days of late summer. 3. They also found that direct applications of abscisic acid to non-dormant buds cause dormancy. 4. A reasonable conclusion is that abscisic acid causes seed dormancy in some species, but not all, seem reasonable because many other compounds and factors such as light, photodormancy, and vernalization, temperature treatment (STLD) are associated with seed dormancy. Preventive role as abscisic acid. It is universal among vascular plants. It is also present in some mosses, algae, bacteria, and most fungi. Abscisic acid chemistry. Structure. Abscisic acid is a 15-carbon compound called sesquiterpenoid. Metabolism. It is synthesized by the mevalonic acid pathway. Early reactions are identical to those of isoprenoids such as gibberellins, sterols, and carotenoids. Small percentage of the abscisic acid in chloroplasts can arise from photochemical or enzymatic destruction of a xanthophyll carotenoids called violaxanthin. Inactivation of abscisic acid can occur in two ways. One is by attachment of glucose to the carboxyl group to form an abscisic acid glucose ester. 2. Another inactivation process is oxidation with oxygen to form phasic acid and dihydrophasic acid. Transport. Transport of abscisic acid occurs readily in both the xylem and phloem and also in parenchyma cells outside vascular bundles without polarity. Place. It is synthesized in chloroplasts and other plastid of leaves under the influence of light. Mode of action. 1 is on the plasma membrane of roots. 2. Another is the inhibition of RNA synthesis, transcription, and 3 is the inhibition of protein synthesis, translation. The root plasma membranes make them more positively charged, increasing the tendency with which excised root tips stick to negatively charged surfaces. This effect is probably involved in the rapid loss of K ions from guard cells, where inhibition of an ATPase is involved, and perhaps in the ability of abscisic acid to rapidly inhibit auxin growth. Interference with the synthesis of proteins and other enzymes could explain long-term effects on growth and development, including the proposed role in the dormancy of seeds and buds and the known inhibition of gibberellin-promoted hydrolase activity in cereal grain seeds.